Hello, my name is Melanie Rutkowski, and I'm an associate professor at the University of Virginia. And today, I'm going to be talking to you about a vignette, an unpublished vignette about how the gut microbiome impacts breast tumor metastasis. And so this story is primarily focused on hormone receptor positive breast cancer. And there have been vast improvements in the outcomes for this disease due to the research that has enabled physicians to more accurately predict which patients will develop metastatic disease versus those that do not, based upon clinical pathological features such as tumor size, tumor grade, and tumor-involved lymph nodes. And this generally will dictate the course and duration of therapy. However, a vast number of individuals will still continue to go on and develop metastatic disease, as indicated by this large proportion of individuals within the metastatic population. And so individuals with hormone receptor positive breast cancer comprise about over 70% of the metastatic breast cancer population. And so really, our lab is interested in this bifurcation where we're trying to understand why some patients, despite receiving similar care, will go on to develop metastatic disease, whereas there are others that do not. And we believe that the TAILOR-X trial really helps to exemplify this idea and demonstrate that it is clinically relevant, because they demonstrated, and this was in the context of racial disparities, that Black women who received similar treatment to white women had almost a 40% increased risk of developing recurrent and metastatic disease, and over a 50% increased risk of breast cancer-related mortality. Importantly, this study did not identify any differences in therapy, really suggesting that there are host-associated factors, either environmental or host-intrinsic, that are contributing to these differences in outcomes. And so, in taking a step back, what do we know about metastatic breast cancer? We know that distal metastatic spread tends to occur early, even before the tumor is diagnosed. And it's driven by crosstalk between that primary tumor environment and these distal sites of metastatic spread. And so we wanted to know, are environmental or host-intrinsic factors contributing to this systemic crosstalk between the primary tumor site and these distal sites of metastatic spread? And we reasoned that the gut microbiome is actually a likely liaison that is contributing to this communication due to its ability to systemically modulate the tissue homeostasis and influence the immune system. And so what is the microbiota? And so it is a collection of millions of microorganisms that are living on and inside our body. They inhabit all of the mucosal surfaces. And so this includes the skin, respiratory tract, urogenital tract, and the gastrointestinal tract. And especially in the GI tract, as you can see here, this individual where all of the barrier surfaces are taken away, and it's just how we would look if we were just the microbes. You can see that the largest biomass is within the gut. And so if we think about these microorganisms, generally they're at about an equal number to our human cells. But if we think about the genome content of these commensals, it outnumbers ours at about a ratio of 1 to 150, really meaning that we are a meta-organism. And so these commensals, they have a symbiotic relationship with us, and generally they colonize without pathology if they're in a healthy state. If we're not taking care of them, then they can lead to long-term and underlying pathologies. And they aid in digestion through the production of metabolites, breakdown of food products, and they act as a first line of defense against pathogens and other diseases. Importantly, they influence the education of the immune system, which is really why we are so interested in understanding this intersection between the gut microbiome and breast cancer outcomes. And so multiple factors can influence the composition or the health of the microbiome, such as a diet high in processed foods and fats, lifestyle full of stress, smoking, alcohol, drug abuse, an environment close to pollution, even long-term exposure to chronic antibiotics. And what that does is that it shifts the equilibrium of the microbiota from one that is healthy and low potential to cause inflammation to one that is unbalanced with a high potential to cause inflammation. And the inflammation occurs through various mechanisms. Generally, it is associated with the release of microbial products that can cause inflammation, the abundance of different cytokines and chemokines that will trigger the infiltration of various immune cells into these different tissue sites. And really what we are interested in are the metabolites that they're producing, because some commensals will produce metabolites that tend to associate with inflammation, while others tend to produce metabolites that dampen inflammation. And so the gut microbiome is actually since long been implicated in breast cancer. And this is because they identified a gene within E. coli and subsequently within microorganisms within the gut that had this enzyme called beta-glucuronidase. And what this enzyme does is it takes estrogens that tend to be conjugated. And when they're conjugated, they go into the gut and they're excreted into the feces. However, these microorganisms can deconjugate these estrogens and continue their circulation within the blood. And it's long been known that these estrogens that are either produced by the ovaries, adrenal gland, and some of the most sort of pathogenic estrogens are from the adipose tissue. And so increased adiposity really tends to associate with poor outcomes in breast cancer due to this increased abundance of estrogens. And so since then, there's been thousands of publications that have really tried to understand how the microbiome influences breast cancer outcomes. However, the challenge, especially with breast cancer research, is there's really, to this date, not been a cohort of individuals where researchers have been able to ask if a pre-existing disruption within the microbiota, not one that is affected by the tumor itself or tumor-associated therapies such as chemotherapy, antibiotics, even immune therapies, impact breast cancer outcomes. And so we wanted to develop a model in order to test this exact question. And so the way that we do this is we use broad-spectrum antibiotics, which in the context of mouse studies, it's more or less like a sledgehammer. And we orally gavage it to the animals for 2 weeks. And that enables the acquisition of antibiotic resistance for Enterococcus species, which tend to be more inflammatory. And so we do that for 14 days, after which we cease the antibiotics, enabling them to clear the system, but also to enable dysbiotic reacquisition of commensal homeostasis. And this tends to be very chaotic and inflammatory, after which we'll initiate tumors. And if we look at the microbiome at this point where we're going to initiate the tumors, we use a diversity measure called the Shannon Diversity Index. And what that does is that it measures more or less the magnitude or the different biodiversity of commensal microorganisms that are within this ecosystem. And you can appreciate that in the context of this dysbiosis, the biodiversity is significantly reduced. And so essentially we've taken this healthy equilibrium and shifted it so that it's enriched in more pathogenic and inflammatory commensals, leading to chronic low-level systemic inflammation. And what we find is that if we look at the composition at this time point, there's actually a lot of resemblance to a recent publication by Laurent Ziffelgil's lab, where they took treatment-naive individuals with breast cancer, and they looked at the microbiome composition and followed that composition and the individuals looking at outcomes. And they found that the enrichment in certain commensals, which are highlighted in the left in red, tended to associate with a higher incidence of metastatic disease, whereas the commensals that are highlighted in blue, that are reduced in our dysbiotic animals, tended to associate with better outcomes, really suggesting that this model does and may have clinical relevance in terms of what the microbiome composition looks like in individuals with a higher risk for metastatic disease. And we initiated breast tumors, and we used a poorly metastatic cell line and a highly metastatic cell line. And in both circumstances, they grew with equal kinetics, which was initially quite disappointing for us. However, we next asked, is the, incidence of metastasis shifted then? Does it affect other metrics of outcomes besides primary tumor growth? And so what we found was indeed that in animals that had the pre-established dysbiosis, there was a significantly higher incidence of tumor cells that were disseminating into the lungs. And we also found at early time points higher numbers of tumor cells within the blood and distal lymph nodes. And so altogether, this data demonstrated that if you establish commensal dysbiosis and then initiate breast tumors, that predisposes these animals to having a higher metastatic burden, regardless of the primary tumor growth kinetics. We next wanted to test whether this dysbiosis had a similar effect in more metastatic or advanced conditions. And so the way that you model that is you take tumor cells and you inject them into the blood, more or less modeling this idea of circulating tumor cells. And what we found is on the survival curve on the left in the red line, the animals with the pre-established dysbiosis died significantly faster than those animals that had a normal microbiota. And in the end, this image on the right, the animals with dysbiosis and without dysbiosis, the mortality was indeed due to lung tumor burden. And so these data demonstrate that the dysbiosis is modulating the lung environment to either enable the recruitment and extravasation of these tumor cells from the blood into the lungs, or it's changing the lung environment so that these tumor cells are better able to survive and grow. And this is something which we are actively investigating. And so we wanted to ask, how does commensal dysbiosis enhance metastasis? And so we focused on this idea of the metabolites. And so we performed untargeted metabolomics, where we took the feces of animals with or without dysbiosis at the time point where we're initiating tumors or after 12 days of tumor initiation. And in both circumstances, as you can see here on this PCA plot on the right, in the red, the animals with dysbiosis had a vast shift within the metabolomic output within their feces, more or less aligning with the changes that we're seeing within the microbiota. When we looked at the different metabolites that were within the feces of animals with dysbiosis, one in particular jumped out at us, which is this quinodeoxycholic acid sulfate, which is a primary bile acid. And so bile acids are very potent signaling molecules. And when we looked at the primary bile acids within the feces, we saw that not only was quinodeoxycholic acid sulfate significantly increased, but so was cholate. And these bile acids can exert profound influences on the immune system and tissue microenvironments if they're dysregulated. And indeed, this is what we are seeing in the context of dysbiosis. Importantly, we found that regardless of tumor status, this disruption in primary bile acid synthesis remained even after cessation of antibiotics 16 days prior. And so at this 12 days after tumor initiation, we're still seeing a significant increase in primary bile acids compared to animals without dysbiosis. So we wanted to ask, do these primary bile acids or bile acids in general influence the ability of these tumor cells to metastasize to distal sites? And so we used a drug called Welchol. Another name for it is colesevelam. And what this drug does is that it binds irreversibly to the bile acid pool that is passing through the gut, enforcing the fact that it's not able to be reabsorbed back into circulation into the liver, and instead it's excreted into the feces. This drug is very well tolerated, and it's often given to individuals to control cholesterol levels, and individuals with disrupted insulin glucose dynamics. And so we used the colesevelam and tested whether or not that affected metastatic dissemination. Importantly, I will add, although I don't have time to show these data, even though we started the animals prior to inducing dysbiosis, the colesevelam did not have any effect in the dysbiosis of the microbiota. It did not impact the microbiome in our hands between either animals with dysbiosis or without. And so really it's a good measure to indicate that we are primarily, any differences that we see are because we're inhibiting and disrupting this increased bile acid synthesis. And so we found that bile acid sequestration, it did not influence primary tumor growth, which was a relief. However, we found that in animals with commensal dysbiosis, it significantly reduced the number of tumor cells that were disseminating using our poorly metastatic breast tumor model. And we used a more aggressive model and we saw similar results. Even though the tumors grew significantly larger, we still were able to reduce the number of metastatic nodules that were found within the lungs in these animals with dysbiosis. Importantly, when we gave the animals this tumor cell line intravenously, we found that feeding them with this bile acid inhibitor significantly reduced their time to death in the animals with commensal dysbiosis. And so altogether, these data suggest that inhibition of bile acids could provide a therapeutic option for individuals that are deemed to be at high risk for developing metastatic disease. We wanted to ask the opposite question then. And so if bile acids are really critical, and because this Welchol targets the entire bile acid pool, we next fed mice with these two primary bile acids, the cholate and the quinodeoxycholic acid, in their food. And we did the same thing. We asked, okay, now they all have normal microbiota. Does feeding them these bile acids enhance their risk for metastatic dissemination? And so similar to all of our other experiments, the bile acids did not impact the primary tumor growth. However, when we measured for tumor cells within the lungs and for metastatic disease, when we inject those tumor cells intravenously, in both circumstances, feeding of the bile acids significantly enhanced the ability of the tumor cells to disseminate and to grow within the lungs if they're injected intravenously. And so altogether, these data demonstrate that bile acids are sufficient for enhancing metastatic dissemination. And so we took a step back and thinking about this idea about crosstalk and how dysbiosis is facilitating crosstalk from the primary tumor microenvironment to these distal sites, because we wanted to understand how the bile acids are facilitating this enhanced metastatic dissemination. And we wanted to ask whether an unhealthy microbiota can actually induce changes in the normal tissue, even before the breast tumor is initiated, so that that tissue is now kind of primed, favoring a more metastatic phenotype. And we demonstrated that in previous studies where we identified that in the context of dysbiosis, there's this chemokine mast cell access that is initiated within normal tissues, tissues without a tumor, and that then leads to enhanced metastatic dissemination. And so if we take away the chemokine CCL2 or the mast cells and we prevent them from degranulating, we find that we're able to disrupt that pathway in animals with dysbiosis. And so we wanted to ask, are the bile acids contributing to these changes within the mammary tissue environment? And so we initiated dysbiosis and we looked within the mammary tissues of mice without a tumor. And so at this time point right here. And what we found was indeed in the animals that were fed the bile acid inhibitor, not only did we reduce the numbers of mast cells, which is the graph on the right, but we saw a corresponding decrease in the chemokine, which is the CCL2, within the entire mammary tissue. And so these data demonstrate then that the bile acids are restructuring the tissue microenvironment. And so in conclusion, what we find is that dysbiosis leads to increased dysregulation of bile acids, and that in turn is driving an upregulation of CCL2 and the accumulation of mast cells within this tissue microenvironment. That then is transforming this tissue so that it is now more susceptible that if a tumor is there, to enable more metastatic seeding in these animals. And so if we block the bile acids using colesevelam, we're essentially able to reverse the transformation of the tissue environment by the microbiome from one that is not susceptible to one that is susceptible, thus effectively reducing metastatic dissemination in the context of breast cancer. And so our lab is actively working to try to understand a little bit more about these pathways in a molecular way. For example, we know that the mast cells, it's not just a numbers game. And so mast cells have a very controversial role in breast cancer. And it's not just the numbers of mast cells that are contributing, but it's also the phenotype. And so we find that if we take mast cells from animals with or without dysbiosis, we sort them out and we put them into mice without mast cells, the mast cells from the mice with dysbiosis still are able to enhance metastatic dissemination of the breast tumor cells, suggesting that the changes within the microbiome are leading to reprogramming also of the mast cells. And so we're working on understanding how these mast cells are different and what it is about them that is changing this tissue microenvironment in hopes of identifying other molecular triggers that are hopefully novel therapeutic targets. And so we finally wanted to understand whether bile acid signaling in the context of breast cancer is clinically relevant. And so we used the TCGA Atlas, and we generated a gene signature list that is associated with bile acid signaling. And so this would kind of recapitulate, a signaling pathway if bile acids are triggering kind of some of these downstream cascades. And we did a rank score, and we took the top expressing the top 15%, which is in the red, and the lower 15%, which is in the blue. And we found that those individuals with a higher gene expression signature associated with bile acid signaling, they had significantly worse outcome, as indicated in the red, compared to the individuals with lower levels. Again, suggesting that primary bile acids could be a molecular target to reduce metastatic spread in individuals with breast cancer. And so in conclusion, we find that commensal dysbiosis enhances metastatic dissemination of hormone receptor positive breast tumors. And this is due to a dysregulation in primary bile acid synthesis. And we find that blockade or inhibition of these bile acids using colesevelam prior to tumor initiation is sufficient to reduce early dissemination and metastatic seeding of the lungs. And that this is happening in part due to modifying the inflammation that is occurring in response to the dysbiosis within the primary tumor microenvironment. Another area within the lab that we are interested in understanding is that bile acids are really at this intersection of inflammation, but also metabolic regulation. And so, as I mentioned earlier, changes within the bile acid pool and this colesevelam are given to individuals with high cholesterol and a disruption in insulin glucose dynamics. And it's very well known in the context of breast cancer that insulin signaling and insulin resistance tend to correspond with worse outcomes. And so we're working to try to decouple these pathways and understand whether it's a linear pathway, whether insulin resistance is somehow associated with this inflammation, or if there are two separate pathways that are converging on the tissue microenvironment to enable metastatic disease. Future studies are also aimed, as I mentioned earlier, at understanding what about the dysbiosis dysbiosis that is affecting these mast cells, and how are the mast cells changing in hopes of identifying other molecular targets that we could inhibit to prevent dysbiosis-induced metastatic spread. We're also interested in understanding, because we see reduced inflammation and mast cell accumulation in the mammary tissues of mice with dysbiosis after we inhibit the bile acids, is it enough to transform that tissue microenvironment so that it's maybe more amenable, for example, to immune therapies? We're also interested in understanding, because we found that dysbiosis enhances the ability of tumor cells to seed and grow within the lungs, how dysbiosis is modifying the lung environment. Is it driven by a similar pathway as our bile acid data would suggest? And if so, are mast cells and the chemokine CCL2 similarly involved, or is there a different molecular pathway at play? Finally, I would like to leave you with this idea that various lifestyle and stressors can negatively impact the microbiota. Some things are unavoidable, but there are things that we can control in order to try to maintain the health of our microbial ecosystem. For example, eating a diet high in fiber with lots of fresh fruits and vegetables, eating a diet in lean meats, and trying to minimize the cholesterol, sugar, processed foods. Trying to minimize stress. All of these factors can reduce this disruption in the microbiota and help to facilitate health in terms of reducing the impact of the microbiome on these distal tissue sites. Hopefully avoiding this effect where an unhealthy microbiome transforms the tissue to support metastasis. And so with that, I would like to thank the individuals in my lab that are primarily responsible for this work. And so the main individual is Audrey Patello, we're getting ready to submit this manuscript for publication. She's done the large majority of this work because she was interested in this intersection between the gut and the mammary tissue. I'd also like to point out both Mika Poblet and Simona Bashgai, who are interested in understanding how the microbiome is changing the lung environment, and also what is unique, or what are the attributes of these mast cells and how are they influencing the tissue microenvironment to facilitate metastatic spread. I'd like to thank Dr. Alcad Fang and Dr. Claire Rosine, who really helped to initiate this project and set the groundwork for Mika, Audrey, and Simona to continue these studies. I'd like to thank my funding from the Susan G Komen Foundation, Breast Cancer Alliance, and of course the NCI, and the support that we receive at the University of Virginia. Thank you also so much for your attention, and the organizers for inviting me to present this work.